just invite you today to have this great talk with both postdoc Carmen Romero and Francesca Garretti from the Icahn School of Medicine, who are presenting their work. And as we always say, science do not stop. And next year, we'll have a lot of seminars coming up regarding microglia, but also have some new work on neuroimmunology as well. Non-zero receptor neurons got barrier protection. And also we share the invitation that if anyone in the audience of both speakers want to present another work, we're really happy to have you here with us. And we thank you, all of you, for joining us in this movement and help us work with this process. And thank you so much. And so I'll give the floor to the speakers today. And thank you so much.
Hello, everybody.
Hi, everyone. I'm Francesca Garretti, and this is Carmen Romero-Molina, and we will be talking to you about a recent review article that we published in Neuron titled Microglial Efferocytosis, diving into the Alzheimer's disease gene pool.
So as background, Alzheimer's disease is a progressive and irreversible neurodegenerative disorder that affects 40 million people worldwide and is predicted to increase its prevalence in the upcoming years, making it an enormous social and economical problem. So Alzheimer's disease symptoms are the known loss of memory, but also difficulty in doing simple tasks or in having elaborated thoughts. It also is accompanied with behavioral changes, and at the end, the individual loses their independence. Apart from the clear macroscopical changes that occur during AD progression, in the microscopical level, we can see an abnormal APP processing and also defective clearance of it. So this is going to lead to a beta accumulation and aggregation, which leads to the formation of extracellular amyloid plaques. On the other hand, the tau protein, essential for microtubules and especially in neurons for axonal transport, phosphorylates, aggregates, and forms the intracellular neurofibrillary tangles. Apart from these proteinopathies, there is also a clear gliosis. The main glial cells in the brain are astrocytes and microglia. Today, we are going to talk about microglia. These are the main innate immune cells in the brain, and they are constantly sensing the environment, and upon any challenge or stimulus, they can adopt different states, which are not well characterized yet, and this is a main challenge in the field. But as a simplification, we can find this is associated microglia, non-responsive microglia, or neurotoxic subtypes. We can divide, genetically speaking, Alzheimer's disease into two subtypes. On the one hand, we have autosomal dominant AD, which comprises for less than 1% of cases and has an early onset. It's caused by mutation in APP and presenilin, so in the APP processing. On the other hand, we find non-Mendelian AD, which has some familial cases, but most of them are sporadic. They go from an early to a late onset, and the APOE genotype is the main genetic risk. Both of them share clinical and pathological features.
So 70% of Alzheimer's disease heritability can be explained by genetic variants, and advances in genetic and genomic methods have helped to identify these variants that are associated with Alzheimer's. A main method is genome-wide association studies. In this method, you test the genetic data between two groups of people that differ phenotypically, in this case, with and without Alzheimer's disease. And you identify the single nucleotide polymorphisms, so variations at a single nucleotide at a position in the genome, to test for differences in allelic frequencies of these variants across the two populations. The genetic data is obtained in two main methods. So in SNP array, which is a microarray that allows for the simultaneous genotyping of up to 1 million SNPs at once, or whole genome or whole exome sequencing. SNP array takes advantage of the fact that SNPs are inherited in chunks. So you can see an area that is in high linkage disequilibrium. So it is very rare for it to recombine. And identifying or genotyping one SNP can allow for the prediction of associated variants within this haplotype with high accuracy based off of the human reference genome sequences. And result of these GWAS studies can be illustrated in a Manhattan plot. This was a Manhattan plot for the most recent AD GWAS that was published earlier this year. And it shows the variants across the human genome that are associated with Alzheimer's disease at varying levels of significance. However, there are issues with AD GWASs, namely the ancestry. GWASs are performed on populations commonly of European descent. And for example, in this most recent GWAS, up to 100,000 AD cases were included in this study. Alternatively, in an African ancestry GWAS of Alzheimer's disease, only 3,000 AD cases were incorporated. And as you can tell, you compare the two different Manhattan plots with a smaller sample size, what you get is smaller genes that are identified. And so not only is the sample size an issue, but also the diversity of those samples that limit the effectiveness of these GWAS studies in identifying loci. So here we had plotted the number of loci that have been identified with the increasing sample size of the most recent GWAS studies. So as you can tell, the Belanges study included the most number of samples and have identified 75 loci associated with Alzheimer's disease. We've plotted the loci in this graph over here. So you can see here the frequency of these variants within the population and their effect on either risk for Alzheimer's disease or protection against Alzheimer's disease. And here you can see that a large majority of the variants are common in the population and, however, don't have such a strong effect on AD risk or protection. What we're missing here are variants that are more rare in the population, but would have a stronger effect on either AD risk or protection. And expanding sample size and the diversity of the samples can enable us to identify variants within this region. Of increasing relevance is also the polygenic risk score. So within each person, you can look at the cumulative effect of both all the risk and the protective variants within one person to assess the total risk a person has for developing Alzheimer's disease. Another caveat of GWAS are haplotypes, as I mentioned earlier. So the SNPs are inherited in large haplotypes or in large sections. However, it is difficult to understand the causal variant that is driving the association with Alzheimer's disease. In addition, and specifically for Alzheimer's disease, a lot of the variants are localized in non-coding regions of the genome. So it is difficult to understand what is the gene that is driving the association with this variant. However, advancements in functional genomics have allowed for the identification of the causal genes driving AD association. So in functional genomics, you couple genetic data with transcriptomic data or looking at chromatin accessibility. So how is this variant associated with different makeups of the chromosomes in which can enable either expression or inhibition of expression, or also what are these in what cell types are these variants enriched for? So in what cell types are they associated with? And as a whole, these methods have been useful for identifying the cell types associated with Alzheimer's disease. In study by our lab they have identified that AD risk are enriched in immune cells. So looking for cell type enrichment in different cell types, you can see that variants are enriched in cell types of the hematopoietic system, so immune cells. This is in sharp contrast with schizophrenia-associated variants, which, as you can see, are largely enriched in cells of the CNS. Another study took human nuclei from different cell types of the brain, so microglia, neurons, oligodendrocytes, and astrocytes, and looked at the promoter and the enhancer regions within these cell types, and looked for variants that are localized in these regions. And what they found was that variants are associated with microglial enhancers and with little enrichment in other cell types. Again, this is in sharp contrast with psychiatric illnesses, in which the distribution of variants and the enrichment is across the board of cell types and promoter and enhancers. So in all, taking all of this information together, what we mostly see in AD-GWAS is that variants are overlap with causal enhancers. These enhancers are cell type specific, and so this region is relevant only in specific cell types. In relation to Alzheimer's disease, you can see this being relevant in microglia and not in neurons. And variations in the enhancer region can either create or remove transcriptional factor binding sites. Altered binding of transcription factors can affect gene expression of genes that are far away from this region and also not identified in locuses in AD-GWAS. And some downstream genes can be affected while others are not. And some genes are causal in microglia and not neurons, but also not just in microglia in general, but in specific states of microglial activation. This is because we see different transcriptomal and morphological states of microglia in Alzheimer's disease. So normally in a wild-type brain, what you see are microglia that are in a homeostatic conformation, and they have these ramified processes. In an AD brain, what you see is that microglia localized to A beta plaques, and they adopt a more amoeboid-like structure. And transcriptomal profiling of these different classes of microglia have identified the different transcriptomal states in AD mouse brains. So what you see are that homeostatic microglia upon different insults and disease states, such as Alzheimer's disease. What you see is the shifting to disease-associated microglial state. And within these, you have the downregulation of homeostatic genes and also upregulation of specific genes, such as ApoE, CD9, CLEC7A. Interestingly, another study looked at macrophages from mice that were fed a high-fat diet and also from adipose tissue from obese individuals, and looked at the transcriptomal profile of these macrophages. And what they found was that the transcriptomal profile of these lipid-associated macrophages closely mirrored that of disease-associated microglia. So there's a convergence of genes that are relevant in DAM and LAM.
So the pathway analysis of these DAM genes and also the pathway analysis of AD risk genes converge in phagocytosis, endocytosis, lipid metabolism, or innate immune response. And these three pathways are not independent causal drivers, but there is a higher order biological process that engloves all of them, which is efferocytosis. So efferocytosis is mainly performed by microglial cells in the brain, and is defined as the process to maintain tissue homeostasis, anti-inflammatory and pro-tissue repair conditions. It's essential because its failure can promote a very chronic inflammation, neurotoxic environment, and even leukodystrophies. So we can divide efferocytosis into 4 steps. The first one will mean the recognition and migration towards the cargo or any signal. So these cargoes are cholesterol-rich cellular waste, which include apoptotic cells, synapses, myelin fragments, dystrophic neurites, or lipoproteins. So microglia is going to be chemio attracted to them, and when it is close enough, it will engulf them using the phagocytic receptors and then developing a signaling cascade. This is followed by the digestion of the cargo, which include the endolysosomal system, the formation of the phagosome, its moderation, and the degradation of the cargo. All this is a real challenge for the cell, so they need to adapt, and this adaptation process include transcriptional changes, and of course they need to deal with the cholesterol-rich cargo, and they are gonna either store cholesterol or afflux it. These steps are also highly metabolically challenging, demanding, so there will be a metabolic adaptation. So in this talk, we are gonna mention some of the genes involved in efferocytosis that had been associated with Alzheimer's disease risk. We want to clarify that these genes, most of them are not exclusively expressed in microglia, but as we said, microglia are the main cells performing efferocytosis in the brain. So as these genes are involved in this process, they are gonna be super important in microglia. And in addition, in some specific examples, genetic studies have shown a direct association of AD risk variants or protective variants with myeloid cells. So to start, we can talk about opsonins, which are molecules that code the apoptotic cells and other substrates to facilitate their recognition and phagocytosis. Here we find ApoE that, as I said before, is the main AD risk gene. So there are two variants that define three different ApoE isoforms, from higher risk being E4 to protective variant being E2. In these experiments, although it's performed in astrocytes, it's predicted that the same is going to happen in microglia. So they show that when they use secreted ApoE, they use conditioned media. And when there is the isoform 4, there is a reduction in the ability of the astrocytes to phagocytose synaptosomes. Clustering is another AD risk gene, which has a function in efferocytosis as a lipoprotein that also mediates opsonization. And in this study, for example, they show how clustering or ApoG can bind to A beta and enhance the capacity of microglial cells to phagocytose A beta. Moving to the receptors, we find here TREM2, which is one of the main targets that are being studied right now, was a potential therapeutic target for AD, and lower expression of TREM2 is linked to a risk. Actually, as you can see here, TREM2 knockout or the risk variants inhibit the transition from the homeostatic to the down state in microglial cells. So TREM2 can bind phosphatidylserine, can bind also other lipid substrates, and even it's been shown to bind A beta. So TREM2 is gonna help in the microglia associated to plaques to engulf myelin, neuropil damage, and all these myelin-rich substrates, and a reduced expression of it is linked to a reduced plaque compaction and an increase in neuropil damage. In addition, TREM2 knockout is linked to an altered signal transduction with a reduced signaling of SYK pathway and of inflammation activation. And this is also linked to an exaggerated calcium release. In this sense, we can link TREM2 with PLCG2. PLCG2 is another AD risk gene, and its knockout has similar effects to the TREM2 knockout because it's going to accelerate calcium release. On the other side of the coin, we have some other receptors which lower expression is actually protective. In this group, we have MS4A. These receptors are not very well characterized, but they are linked to TREM2 and associated with soluble TREM2, which might be an AD biomarker. We also have the reduction of Don-Itme signals, for example, a reduction of expression of CD33, of PLRA, or of Siglec-11. These are both, all of them converging in the signaling pathway of another AD risk gene, which is INPP5D, encoding for the SHIP protein. So this protein is gonna inhibit BLNK, which is another AD risk gene. This BLNK facilitates the migration of PLCG2 to TREM2. So we're going to have that the TREM2 PLCG2 receptor signaling pathways, higher expression is going to be protective. And on the other hand, a lower signal of the Don-Itme pathways, if it's lower expression reduction, is going to be protective as well. All these steps, define me and needs me, need a cytoskeleton rearrangement. And in this step, we can find a lot of AD risk genes. I'm going to mention ED, which encodes for a protein which forms part of the polycomb repressive complex 2, and it's involved in immune receptor mediated signaling. And also AB3. AB3 has been shown a long time ago to be involved in efferocytosis in nematodes. And of course, it will be important in efferocytosis in microglia. So AB3 knockout mice show an increase in the A-beta plaque content. And AB3 has recently been proposed as an early biomarker of Alzheimer's disease.
So following substrate recognition, microglia extend the plasma membrane around the target and eventually fuse to form a phagosome. A phagosome then matures through fusion with other vesicles to gain the ability to digest cargo. This pathway is the endolysosomal pathway, and it's a network of intracellular membranous organelles that are interconnected in a dynamic fashion. The main function of this pathway is to digest, to recycle, and to eliminate internalized or intracellular material. And GWAS studies have identified several associations between endolysosomal system genes and AD risk. One of them is BIN1. BIN1 is the second strongest risk factor for Alzheimer's disease after ApoE, and it functions in membrane remodeling in the endolysosomal system. BIN1 encodes for the protein bridging integrator 1. So one study has highlighted this region of the BIN1 promoter, which is shared across all different cell types within the brain. However, the AD-associated variant is located in this pink area, which is the microglial specific enhancer, which is relevant only in microglia. When you remove this microglial enhancer that is containing the AD-associated SNP in iPSCs, and then differentiate these iPSCs in different cell types, you can see that it affects BIN1 expression only in microglia, but not in other cell types. So this is a great example of how a variant localized in a non-coding region, which is common in Alzheimer's disease, has an effect only on specific cell types, and in this case, microglia. When BIN1 expression is decreased in microglia, you see a defect in the phagocytic capacity of microglia, both in an unchallenged and in a challenged state with LPS. Another gene that is associated with the endolysosomal system is SORL1. And common variants are consistently associated with AD and large GWAS. However, there are also some rare coding variants that cause truncations or missense mutations that have been reported in familial and sporadic AD cases. SORL1 encodes for sortilin-related receptor, which is part of the VPS domain receptor family. And SORL1 knockout or SORL1 mutant microglia, so this mutant impairs cargo binding and recognition. Looking at the proteins that are expressed in these microglia, you see an upregulation of proteins in the early endosome pathway, while also downregulation of lysosomal proteins. When these microglia are then xenotransplanted into AD mice, you see a downregulation of the number of plaque-associated microglia, in addition to a reduction in the phagocytic capacity of these SORL1 knockout microglia. Another gene is GRN, which encodes for progranulin. Progranulin is a secreted pleiotropic growth factor that is a key regulator in lysosomal function. GRN knockout mice display microglial hyperactivation and also a damped transcriptional profile, but also a lysosomal dysfunction shown here with increasing levels of cathepsin D and also increasing amounts of lipofuscin. In addition, you have lipid dysregulation, and you can see here that in GRN knockout mouse, you have a decrease in different classes of lipids. When granulin is knocked out in mouse macrophages, these macrophages were then treated with fluorescent apoptotic cells, and there's an increase in the capacity for these progranulin knockout macrophages to phagocytose apoptotic cells.
So as we were saying before, all this process is going to be very demanding for the cell. So you need to adapt and deal with the cargo at the same time that coordinates an anti-inflammatory and pro-tissue repair response. So the transcriptomal changes that occur upon efferocytosis include PU1, which is an AD gene, and lower expression in myeloid cells delays the age at onset of AD. PU1 is a master regulator, and it determines the differentiation of myeloid cells. So researching and performing experiments of PU1 knockout is challenging, because if you need to differentiate the iPS cell, the human-induced pluripotent stem cells, to microglial cells, you cannot knock out PU1, but you need to create a conditional knockout. PU1 is going to regulate the expression of several AD genes, and our lab has shown that a reduction in PU1 expression polarizes the cells towards a dam-like state. Also, by others, it's been shown that a reduction in PU1 is going to promote the polarization of microglia towards IL-33 dam states. So apart from these master regulators, we have also lineage-specific transcriptional factor and also broadly expressed signal-dependent transcriptional factors. In this group, we find 2AD genes and R1H3 and 2, both encodes for ALXR nuclear receptors, which are involved in the regulation of cholesterol efflux. The knockout of either an R1H3 or ApoE or AFCA1 impairs the cholesterol efflux, and it's going to compromise the efferocytosis of myelin debris and remyelination in most models of demyelination. On the other side, ALXR agonist will rescue microglial lipid dyshomeostasis both in ApoE and TREM2 knockout mice. As a transcription factor, we also have EED that I mentioned before. that is gonna form part of the PRC2 complex, which is gonna modulate the methylation of histone 3, and it's gonna be a gene repressive factor. So it's been shown that microglia with EED deficiency display an enrichment in apoptotic cell clearance, cholesterol response to lipid, and so in general of the efferocytosis pathway.
So when microglia ingest lipid-rich debris, the excess cholesterol is either esterified for storage into lipid droplets, which then forms foam cells, or it is effluxed out of the cell to acceptors such as ApoE. This is to prevent the toxic accumulation of free cholesterol in the ER and the formation of cholesterol crystals within lysosomes. ApoE is an acceptor of this cholesterol, and it binds to cholesterol and other lipids to form lipoproteins. As Carmen had mentioned, there are 3 different isoforms of ApoE, and these are determined by 2 different variants. These variants cause a conformational change in ApoE4, which has been shown to render ApoE4 as a less potent acceptor of cholesterol. Here you can see that in ApoE4 microglia compared to ApoE3 microglia, you have a reduction of cholesterol efflux. In addition, within these cells, in ApoE4 microglia, you can see that there's an increase of cholesterol biosynthesis. So in ApoE4 microglia, you have the double hit of decreased cholesterol efflux in addition to increased cholesterol biosynthesis, which renders ApoE4 microglia cholesterol rich. And another AD risk gene are ABCA1 and its close homologue ABCA7, which have been identified in several GWAS studies. They bind to phospholipids and cholesterol and bring it to ApoE to form lipoproteins. ABCA1's role in cholesterol efflux is quite well established. However, ABCA7's role in this pathway might be due more to its ability to flip phosphatidylserine from the inner to the outer leaflet. This function, flipping phosphatidylserine from the inner to the outer leaflet, occurs during apoptosis. So it signals to neighboring cells that this is a dying cell that needs to be phagocytosed. And here, when you see that liposomes have been reconstituted with phospholipids of different classes, you see that the ABCA7's enzyme activity is at its best when the liposomes are reconstituted with phosphatidylserine. And looking at ABCA1 and ABCA7's close homologues in C. elegans worms, you can see that only in ABCA7 deficient C. elegans, you have the accumulation of apoptotic cells stained here with an X and V. And this is not due to increased apoptosis, but rather a decrease in the phagocytic capacity of apoptotic cells.
So last, we are going to talk about genes involved in microglial metabolism. It's been published that TREM2 is essential for microglial metabolic fitness through the mTOR axis. So as you can see here by a seahorse experiment, there is an important decrease in maximal respiration of microglial cells when we have any risk variant in TREM2. We would also like to mention a new AD risk genes that has been recently identified as AD risk gene in a proteomic study. So lactamase B, it's mitochondrial gene, which has never been studied actually in myeloid cells, or at least in microglia in the AD context. However, the cancer studies point that LAC-B is essential for the oxidative phosphorylation, the lipid metabolism, and it's going to determine the ability of the cells to proliferate, differentiate, and migrate, which of course are essential capacities of microglial cells. We also have in the mitochondria OPA3. So OPA3 is being shown to be involved in mitochondrial hyperfusion. Hyperfusion is an adaptation, a dynamic adaptation of mitochondria under increased oxidative phosphorylation or mild stress or nutrient starvation. As you can see in this electron microscopy picture, in APP PS1 mice at advanced age, like 8 months old, there is a mitochondrial elongation, which points that OP3 may be an interesting target. So how all this coupled together, how can microglia efferocytosis modulate AD risk? On the one hand, microglia needs to effectively recognize only like unwanted or damaged synapses, so and phagocytose them. So although the pruning mechanism is much more studied in development than in adulthood, it's been shown that the mechanism of pruning and synapse engulfment because of damage are similar. And we need, as I was saying, microglia to selectively identify only unwanted synapse. On the other hand, microglia needs to efferocytose AD-related debris in the context of the plaque, of the neuropil damage, all the cellular debris, and it's been shown that microglia plays an important role in myelin homeostasis. So the failure of the efferocytosis process is going to lead to an accumulation of debris, pro-inflammatory molecules in anti-repair environment. As we have been showing, AD genetics suggests that higher expression of genes that promote efferocytosis and lower of the one which inhibited, it's gonna be protective, it's gonna decrease AD risk. So all this state-of-the-art technology has allowed us to identify several losses associated to AD risk and even to map the loci to causal genes. However, much more effort is required to really understand how these causal genes are going to modulate AD risk, which are the causal pathways. So for that, we and others use smaller interfering RNA, RNA or CRISPR technology to modify cell lines, primary microglia, or human-induced pluripotent cells that they are gonna then be differentiated to microglia. We use them in monoculture, or as we really want to study the interaction with other cell types, we use organoids and other 3D system. If we want to move in vivo, but keep the human genetic, very novel and innovative technique is to xenotransplant this iPSC into mouse, and then they will differentiate into microglia. And of course, we can use genetically engineered mice. All of this is going to allow us to study microglial function, how it interact with surrounding cells, and at the end, how it's going to influence the global physiology. So far, there are several preclinical and clinical trials for the promotion of TREM2 and PLCG2 for the inhibition of MS4A, CD33, or P1, and to mimic the protective ApoE isoform. So as a take-home message, we would like to say that this concept of the polygenic risk score of each individual is going to contribute to an improved prediction of the disease risk and the rate of cognitive decline. And it's going to allow us to better design clinical trial and even aim for the development of preventive therapeutics. With that, we would like to thank the co-authors of this paper. Of course, thank to all of you for your attention, and we are happy to take questions.
Thanks, Carmen. Thank you. Thank you, Francesca, for very extensive introduction of Alzheimer's disease and the role of microglia. Now we'll welcome anyone who has questions to unmute yourself, or you can type in the chat. Let's just give them a couple minutes. Carmen and Francisca are the nicest person on the whole world. So if you have questions, don't be afraid. And
Hey,
okay,
can I ask a question?
Of course.
Hey, okay, thank you. Nice to meet you. It was a pleasure hearing your presentation. I'm really interested in microglial metabolism, and that's basically what I'm trying to address during my PhD thesis. I was wondering whether there is a way to leverage those lipid droplets that normally has been described in H microglia, for example, the Tony Wiss-Korai paper. I think my question is basically, is there any way we can leverage those lipid droplets that form across like across adulthood and that are even more noticeable during like disease?
So I guess that's promoting all the mechanism that Francesca mentioned during the cluster afflux are gonna induce that, right?
So if you mean by modeling a lipid droplet formation, you can create foam cells within microglia by treating them with different lipids to kind of induce this pathway and induce the accumulation of lipids. So you can either treat them with oleic acid or palmitate or other different fatty acids, which will be taken up by microglia and induce foam cell formation due to the accumulation of lipid droplets. And that would be a great way to then couple that with what you were interested in studying and seeing how the metabolic function is impacted.
I think it's even going a little bit beyond. I'm sorry that I'm taking so too many minutes. It's more like taking advantage of those lipid droplets, because I think I remember a paper was recently published about the exokinase 2, and by ablating exokinase 2 in microglia, it was possible to improve lipid metabolism by the upregulation of LPL. I think it's lipid proteolipase. So I was thinking on a way that it could be possible, even though microglia might be like dysfunctional because of this lipid droplet formation, still a way to sort of rescue microglia functions.
Yes, definitely.
Yeah, I guess the promotion of the genes that we already know that are involved in the pathway, if you want to try to overexpress them and see the phenotype, if they can rescue the lipid droplet accumulation.
Thank you. Thank you for the presentation as well.
Thank you.
And we have one more question in the chat. Does this suggest that games are protective?
So that's a little bit of the $1 million question, but what we see if you inhibit them, you actually have fewer plaque-associated microglia and actually a worsening of an AD phenotype in AD mice. However, I think it has to do also with the timing in which these disease-associated microglia genes are upregulated. That is also important in fine-tuning the response and either protecting or increasing risk for AD-associated pathology.
I think also that in the case of DAM microglia, their action towards A beta and tau pathology is a little bit antagonic. So at least in my experience working on Alzheimer's disease, when you reduce microglial DAM phenotype, or let's say activation to tau more broadly, they are less able to handle A beta pathology, and this can worsen the animal condition in this case. However, if you are talking about a tauopathy model, that we all know that these P301S mice don't really represent AD because they have excessive amounts of tau. In that case, what I've seen is that reduced microglial activation is actually beneficial for the tau pathology progression. So you can, I guess it's gonna depend as always in the stage at which you talk and which is the most prevalent pathology, which cerebral region. So as what Francesca was saying, this is a very tricky question. I don't think there is gonna be a definite answer. And what we can aim to do is more than saying DAM or non-DAM. Try to really define the different microglial states and modulate each of one independently, trying to be more precise.
Do we have any more questions? Okay, then let's give the biggest applause to Carmen and Francesca for fantastic presentation. And if we have any more questions, that we will email you and follow up with you. And thank you all for joining us the last seminar of 2022, and see you guys next year. Thank you.
Thank you very much. Thank you. Thank you.